Assistant Professor, Department of Biotechnology, to welcome Dr. Prashant S. Zaki, sir, as a token of gratitude. Thank you, sir. Now I request Dr. Rahul Kamble, Assistant Professor, Department of Botany, to welcome Dr. Kanaya M. Daduri as a token of gratitude. Thank you, sir. So to deliver the first oral presentation, I call upon Mr. Virendra Kishore Sangode, Assistant Professor, M.B. Patel College of Arts, Commerce and Science.
Hello.
So please note that you have 8 minutes of presentation time and 2 minutes for discussion.
Shall I start?
Yes, sir.
Very good afternoon to all of you. I pay a very high respect to Tathagat Gautam Buddha and Dr. Babasaheb Ambedkar. Apart from that, I would like to today's my presentation is about WVC, Wildlife Vehicle Collision Data, a potential tool for road ecology research and biodiversity. That is a conservation part that is to be done in and around Gondia district. Next. Now, this topic is basically based on the roadkill. When we think about the term RWVC, that is the WVC, Wildlife Collision Data, what do you think? What is it relates about? It totally relates upon the kinds of vehicle which we draw, which usually we drive on the highways. Think about a car driving the speed of more than 100, 120 or more than that. Probably, with respect to that term, we all know that roadkill, although the word seems to be quite new now in the dictionary, but whenever we do observe or report, it is quite totally heartbroken to see the car cases of animals, isn't it? Whenever we are driving fast, we can see the animals which were totally quite a mismatch, very brutally killed. Meanwhile, so what is the basic reason? The reason is that almost this data initiates, we are having a protected areas comprising 5% of Indian geographical expanses, of which when we compare the comparison of animals, we do find that 7.6% of almost mammals are over there, 12.6% of avifauna, 6.2% of herpetofauna, and there are many more inhabiting species. So our study comprises an in-depth study which are having two objectives. First, it would be a collection of data with respect to opportunistic or random observation that usually be done. Basically, it needs a time. We have to stop our vehicle, we have to get down, we have to look after the which kind of animal is over there. Followed by, it can be a transect or target in-route study. When we think about transect or target in-route study, it comprises one complete route that we have to track on. Say for example, I used to track from Nagpur to Sadaka Arjuni, which comprises two particular national forest areas like Navegaon, Nagzira, followed by Koka Wildlife Sanctuary. Now during that observation, I do observe there are three parameters which are in concern with the wildlife protection. It can be traffic characteristics, use of land, road infrastructure, animal behavior. So basically these are three instincts that animal do have a pattern of following. Now, as a part of study, whenever we are driving quite with a very high speed, it is the mindless driving, and our data is quite designed to note the practical observation of animal, whether it is quite alive, dead, or what is the exact situation, and based upon that, we are reporting to the forest officials. It's a result over here. It's the first picture which I located at National Highway 6, Jamli, Sakoli district, Bandara. It's a roll kill counter 1. You can see the picture of what? It's what? It's a Indian python having scientific name Python molurus. Another data, it's a roll kill counter of checkered killback having scientific name Xenocrophis piscicator. The third one, it's what? It's a wild boar having scientific name Strophas cristatus. Another picture, endangered animal one. Again, it's a Canopus lupus palpus. We can call it as a wolf. Another one, it's a fox, isn't it? So what do you think, whether they are quite important to us or not? Definitely they are quite important to us, as if apart from human life, we have to also think about the animals which we can do observe in our nature. For to observe them, we usually pay lots of money going Tadoba, going Animo national parks and all. But apart from this, if you do pay attention to the road kills, it will be quite beneficial to conserve. These are the references. And just a quiet please to all, drive slow and let others live happily. Thank you very much. If you are having, can ask.
If anyone from audience want to ask any question, just ask 1 or 2 questions.
Is it okay? Thank you.
Next, please.
Before we proceed to the next oral presenter, I request Dr. Devraj R. Saxena, Head of the Department Zoology from Kamala Nehru College, Nagpur, to kindly be seated on the dais. Thank you, sir. Now I request Dr. Ashwini Aswal, Assistant Professor, Department of Botany, to welcome Dr. Devraj R. Saxena as a token of gratitude. Thank you, ma'am. Thank you, sir. So we are proceeding with the next oral presenter, Sayyeda Parveen Qureshi. Ms. Sayyeda Parveen Qureshi. Okay. Next, Naina Sudhakar Shirbhate. Naina Sudhakar Shirbhate. Okay. Dr. Padmavati Srinivas Rao. Madhukar Fakirazi Zado. Ravindra Balkrishna Zode. Veena Mahendra Mahajan. Yashwant Agde. Sunil Akare. Vaishali Jamgade. Mr. Jagannath Gadpaile. He is Assistant Professor from S.N. Moore College of Arts and Commerce.
Hello. Good afternoon, everyone. Respected juries, I am Dr. J.V. Gadpaile from S.N. Moore College, Bhandara, Tumsar. And today's my presentation topic is floral study of Alicicarpus species from Bhandara district. So this genus is the very well-known genus which is mostly occur in along the roadside. You can find this plant in the agriculture field and in wasteland. Most of the student also observe all these specimens, but they doesn't consider which are the species and whatever the characteristics are found in that particular specimen. So I do my work as a taxonomist to explore the Alicicarpus species from Bhandara district. So I would like to present my presentation. So this is the study area, Bhandara district, satellite map as well as in Maharashtra state where it is located, Bhandara district. I just go there, and these are the sites where collect I the specimens regarding Alicicarpus species. So I found that these species I collected from 2020 to 2022, almost 2 years or 3 years survey, and that work is just compiled or considered for the presentation. Brief introduction regarding the Alicicarpus species is that overall this plant belongs to family tribe Desmodiaceae, family Fabaceae. Worldwide, 35 species are there and 20 intraspecies found worldwide. In India, it is represented by 18 species and along with 9 varieties. Out of them, 8 species and 4 varieties are endemic to Maharashtra. That is given by the honorary Pokde sir, Dr. Pokde sir. And after that, in Maharashtra, it is represented by 70 species along with 11 varieties. The present work represent the complete pictorial illustration or the floristic characteristic of this genus. And in this my study, I found that there are 13 species which is collected from the Bhandara district. Previously, there is a 6 species reported only. Now I am going to add 7 species to the Bhandara district as a new report. So these are the characteristics when we consider the floral characteristic of Alicecarpus. So there are 3 kind of different inflorescence, that is lax, compact and elongate. After that, these are the species where I found that there are different colors of flower, means monocolored and bicolor flowers. So the flowers which are showing only single color, they are monocolored, and the flowers which have the 2 color, they are considered as a bicolored. So these are the species where the bicolor flower and the single monocolor flowers are found. These are the pictures. These are monocolored. These are bicolored pictures of the flowers. This is the important characteristic when we identify the Alicecarpus. There is the presence of bracts and they show the different size, shape, variation along with the overall dimension of the bracts. So these are the bracts. There are primary, secondary bracts, and all are the most important characteristic in Alicecarpus species. Calyx. This is again the important characteristic during the identification, that is glumaceous calyx, scaraceous and membranous calyx. These different. On the basis of that, almost 8 species you can differentiate of the Alicecarpus species. These are the floral characteristics regarding bracts, calyx, standard and the different floral characterization. Another one, this is important characteristic, which are the pods found in Alicecarpus species. So when we consider the morphological characteristic of pods of Alicecarpus, then we found that mostly there are 3 kind of pods are found, or legumes. One is noded, second one moniliform, then third one that is beaded. Mostly we found these 2, that is these are the 2 forms of the pods mostly found in Alicecarpus, and these are the distinct one. If we find the segments in the Alicecarpus, whether it is different or you can say 4, 5, 6, or that may be a key characteristic for the identification of Alicecarpus species. These are the orientation. When the first layer of the pod we open, then we found that the seed morphological orientation or the arrangement of these, that is the smooth, this one is the smooth, then transversely striated, reticulate and transversely rugose. These are the seed characteristic, and here we found that there is a 2 characteristic, that is the this one is the hilum, and then the upper portion of the hilum, which kind of color is found there. Okay, if that color is different, then you can consider that may be a variation or that may be a new chance for the new species. These are the species when I wander throughout the Bhandara district and collect all these specimens. That is Alicecarpus longifolius variety major. You can find out all these species. Another species, they are enumerated and presented in upcoming my paper also. Thank you very much. I would like to thank you my mentors as well as my guide, and those are the members who helped me during the identification. Pokde sir and Professor Milind Sardesai sir from Savitribai Phule University, who helped me for the identification, and I extend my thanks to the organizers to give the opportunity to present my work. Thank you.
Hello. Now session is open for questions. So please audience, you can ask the questions. If any questions, please. Can I ask you, sir, one single question? Okay, so those species you are discover, it is generally it is found in that district, or it is found in other district also?
Only 2 species. They are mostly found in Bhandara district throughout the Maharashtra state. One is Nikeana and another one is Pokrianthus. These two species reported earlier there in Western Ghat and Aurangabad region. Nowadays, we collect that species from Bandara district. So, dominancy of that species mostly occur in Bandara district. Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir. Now I call upon the next oral presenter, Ms. Shruti Gharade. Ms. Shruti Gharade. Not there. Okay, next, Ms. Anisha Rahate and Ms. Diksha Daima. The students of BSc Biotechnology, Dr. Ambedkar College, Nagpur.
Hello, आवाज आ रही है?
Hello. Good afternoon, everyone present here. My name is Anisha Rahate.
And my name is Diksha Daima.
We are the students of first year from Department of Biochemistry and Biotechnology, Dr. Ambedkar College. Today we are standing here to present our study on cytogenotoxic effects of plastics using allium cepa as a model system. Imagine you all are standing in a luscious green field with a beautiful scenery, and suddenly out of nowhere it starts raining plastics. It sounds quite absurd, right? But at present it's like this condition is not impossible to happen. Just as water is present everywhere on our planet, similarly plastics can now be found in every single biosphere. Due to its low cost and convenience factor, plastics have now become a major cause of concern.
Do you know, according to a recent survey conducted by National Productivity Council, around 20 meganewton tons of plastic was consumed alone by a country in 2020. And estimations say that it may rise up to 20 kg per capita in 2022. Here is the given data of which sector uses how much plastic. So, but here if we focus on one major part, we can see that 80% of plastic is consumed by packaging sector alone.
Harmful effects of plastic. We all are very much aware what plastic can do to the living world. Let's get a brief about this. Plastics contain what we call the EDCs, or the endocrine disrupting chemicals, which mimic, block or interfere with the hormones of the endocrine system, and they may lead to a diverse array of health issues. As Diksha said in her earlier slide, with increasing rates of plastic production, the rates of acute and chronic diseases and deaths arising due to the exposure to EDCs is increasing day by day.
As mentioned earlier, these EDCs contain one such harmful chemical called BPA. BPA stands for bisphenol A and is in use for making plastics since 1950s. Polycarbonate plastics and epoxy resins contain this BPA, and generally these polycarbonate plastics are used by the packaging industry to store foods and beverages. These foods and beverages are more prone to the process of leaching and henceforth release of BPA and other harmful chemicals into them. The question that stands here in front of us is, does anyone not really know about this?
Everyone are indeed aware about this in some or the other way. Consumer gets information regarding products from news and social media. But with so many warning signs surrounding us, finding danger in these potentially harmful chemicals is almost very hard to believe, because not every warning is true. So here we must possess the proper conscience to judge between which warning is true and which is not.
Now let us see the effects of BPA on human body. This can be directly correlated with carcinogenicity. This BPA mimics as estrogen to interact with the estrogen receptors alpha and beta, and henceforth causing cell proliferation, apoptosis, or even its migration, thereby causing cancer promotion. Investigations said that maximum content of BPA shows the amount of breast cancer, prostate cancer, thyroid cancer, and various types of cancers. And henceforth we can here anticipate that BPA is a probable human carcinogen.
BPA can also be associated with disruption in fertility. It is said to enter the woman's body and trick the reproductive system into believing that it's a hormone. This blocks or changes what the body's natural hormone is actually supposed to do, and it is shown to cause poor egg development, as well as it may also alter the DNA of the fetus, leading to certain birth defects. Studies have also shown that BPA can also cause many reproductive issues in males as well.
Other than that, BPO also affect various organ systems of the body too. Now let's move on to the allium-SEPA test. The allium-SEPA test is basically mainly used as a bioindicator. The allium-SEPA test is one of the few direct methods to estimate the damages in the system that are caused due to potential mutagens and carcinogens. In this test, we can see that these are the materials required. Many of the scientists have provided research papers that BPA causes cancer, but there are not even that much articles available on the internet that shows us that BPA and its in vivo cytogenotoxicity
Are present in our day-to-day products as well.
Hence, we use 3 samples which are used in our day-to-day life. In case of sample 1, we boil the water at different temperatures and pour them into different cups. While in case of sample 2 and sample 3, we cut them into small pieces, boil them at different temperature and shifted the extract into different containers. And henceforth, thereafter, we let the onion root tips grow on them.
We let the allium-SEPA grow for around 24 hours in these water samples. Different morphological, cytological and chromosomal aberrations were seen. In case of morphological aberration, variation in root length and appearance can be seen at different temperatures. After this, we stain the root tips and observe them under the microscope.
We observed that with increase in temperature, the bleaching process kept on increasing, and this increase in BPA results in significantly reduction of mitotic index. And here is the slide which shows us the normal mitotic stages for reference.
As we said earlier, BPM can also cause chromosomal aberrations. So these are the different conditions which we can see in BPA affected cells. Here if we take an example of lagging cell, so this condition is observed because of the formation of dicentric chromosome due to the presence of 2 centromeres. Another example which we can take is of anaphase bridge formation, which is a chromosomal segregation error which we find during mitosis.
We also observe some cytological aberrations. Here if we see micronucleated cells and binucleated cells, generally these types of cells are cancer causing cells. In micronucleated cells, as they are very small, they are unable to express the genome. While in binucleated cells, they have 2 nuclei.
Conclusion. BPA has been accumulating in our body since a very long time. Comparing it to the allium-SEPA cells, now we can see what BPA can do to our cells as well. From our experiment, we can see that our day-to-day commodities like plastic plates and cups, which we initially used to think are pretty harmless to us, can actually harm the environment and humans in the worst way possible.
While doing our research, we came across this fact. Isn't it a matter of concern that even our future generations are not so safe because of this BPA? It's a high time to rethink right now, because not today, then we are definitely endangering our future. Henceforth, we are trying to find various alternatives for plastic as a whole.
We are in dire needs of alternatives to completely replace plastics and at the same time not harm the environment as well. So while doing our research, we came across some such alternatives, namely compostable plastic, bioplastics and hemp plastics. These are non-toxic and biodegradable in nature, and they possess very similar properties to traditional plastics. So if we use them efficiently, we can completely replace plastics using them.
And here we are concluding our part. I would like to thank our teachers, Mr. Pradeep Hirapure sir, Shweta Paransme ma'am, Rita Lakukal ma'am, to help us and guide us through this. And I hope that smiles like of this child may stay in our future. Thank you everyone present here for lending your ear.
Is there any question from audience? So both of you have taken very good topic. I have just one question. During your experiment, how much time you have kept water in that plastic containers?
Sir, for around 24 hours.
Then we are normally using different type of plastic containers either for tea or for other juice or any food material. And if such a type of results observed cytologically in a just few minutes or hour in a plastic container in case of bulbs of onion, then what happens to human body?
Sir, as in our conclusion part, we said that BPA has been accumulating slowly since a very long time in our body. So it does happen in few minutes as well, but we tried to do our experiment in the very extreme conditions so that so this way we can observe the most strong aberrations in the cells.
Okay, you have accused good problem. Okay, thank you.
Thank you,
Just a minute,
Anisha
just a
and
minute.
Diksha.
There is one more question from.
Hello, I would like to know what do you mean by apoptosis? Apoptosis?
Sir, it's a condition where the cell dies. It is like stages. Sir, actually we are like trying to study. We are in the process of studying. So it is a conditions in which the cell dies. It has particular levels and stages it goes through.
Okay.
Thank you, sir.
Thank you, Anisha and Diksha. We move on to the next oral presenter, Miss Anita Kadkai. Miss Pooja Nanavari. Next. Mr. Ramdas Atram. Asiya Nasin. Miss Sakshi Ghule.
Good morning, one and all. Myself, Sakshi Gule. I am pursuing my MSc Botany in MSc Botany from Institute of Science, and I am present here to represent my topic: Survey of Wild and Cultivated Vegetables from Brahmapuri Taluka of Chandrapur district from Maharashtra, with reference to utilization. Yes, समोर नाही जाऊ. Introduction. Since ancient times, the local people dependent on plant for fulfilling their food requirement for their livelihood. So the local people used wild vegetable plants for their utilization. But during the race of civilization, human beings started farming, so turned their attention toward the cultivated plant species. But still, local people use the wild vegetables. Different types of vegetables are source of food material. Besides this, local people believe that some of the seasonal wild vegetable plants are good for health and help in curing various diseases. These vegetables have nutritional value. Traditionally, the leafy vegetables are more nutritional. Wild vegetable plant refer to species that are neither cultivated nor domesticated but available from their natural habitat and used as a source of food material. Vegetable plays a very important role in human life. They have been used for food, medicine, fibers and other purposes, and also as a fodder for domestic animals. Then material and method. For present study, various villages from Brahmapuri Taluka were selected. The investigation was carried out from rural areas and vegetable market in Brahmapuri. During the investigation, regular visits were made two different villages from Brahmapuri Taluka. Information were collected from visits and interaction compiled in tabulated form. Group discussion was also conducted with the local knowledgeable persons and data of interaction compiled in the tabulated form. The diverse information about local names, plant part usage, recipes and their utilization was also inquired. Observation. In the present study, 45 plants used as a vegetable by local community has been studied, which belongs to 29 families of 45 plants belong to 29 families of angiosperm. 28 are dicot and 15 are monocot. This is the table, and here are the 45 families mentioned in the table. These are some of the plates which I have taken from Brahmapuri Taluka. Figures: location of Chandrapur district, number of monocot and dicot plants including one fungi, proportion of wild and cultivated vegetables, herb, shrub, tree and climber proportion, number and percentage of different plant part used as vegetables, representation of number of families. Result and discussion. In present study, 28 leafy vegetable has been recorded. Chauhan et al. Also studied 51 leafy vegetables from Chhattisgarh, Amravati. Dani et al., Deshpande et al., Dobre et al., Bhogaonkar and Nimbekar has also published various research papers on wild vegetables and their usage for local communities from Satara, Amravati, Gadchiroli and from Chhattisgarh states. Some of the vegetables from study area are also used for same purpose, but the method of preparation is different in different areas. In the present study, it has been observed that local people have good deal of knowledge regarding the wild vegetable, and the root, stem, leaf, tender leaves like plant part used for the purpose of vegetables, preparation, recipes, mode of utilization, different in different communities. Some of these vegetables have also other utilitarian values to the local people that are used also in medicinal purpose. Awareness regarding cultivation and utilization of this vegetable is also required among high society and urban population. Acknowledgement. Thank you.
This audience, please ask the questions. No any questions? I think after lunch session, everyone just slow down. Huh? Nobody want to ask question. Okay, the one question I want to ask you. Any medicinal application of this plant?
Which plant? Anyone?
No. Those you explain in your slides. All these are the explanations.
Moringa oleifera.
No. I want to medicinal applications.
Medicinal application.
Capparis
Capparis zeylanica is used as a medicinal.
It's okay.
Next.
Thank you, Sakshi. Next, Miss Supriya Gedam. Miss Supriya Gedam. Mr. K. Narayanan. Miss Renuka Lonkar.
Good afternoon, everyone, respected juries, delegates, and my fellow researcher. My name is Renuka Arvind Lonkar. I'm from Sevadal Mahila Mahavidyalaya. My topic name is Preliminary Study of Mollusca of Shivapur Lake, Umred, District Nagpur. This is my study area. It is situated at Umred Bhivapur Road. It is 4 kilometers at the north of that particular road. It is a very vast lake. It is a very big lake with lots and lots of biodiversity of all kinds of life forms. This is basically the view of Shivapur Lake, as you can see, very big. These are the major freshwater mollusks I found in that lake. 5 species of gastropods were found and 3 species of bivalves were found. This is the list. Basically, first one is Bellamia bengalensis. It is from subfamily Bellamidae. First, the phylum Mollusca. Mollusca means the soft-bodied organism. The characteristics of this particular phylum is during development, they undergo torsion, that is a complete 180 degree of transference of or rotation of their visceral mass. Therefore, their anus is situated more or less above their own heads. This is the particular photo of Bellamia bengalensis. It is an invasive species. It is used for studies and for consumption also. Another species is Thyra scaba. This is the photo. This is basically a trumpet snail. It is, as you can say, it is a very small one. It attaches itself to stones or basically small particles in the lake or plants and other stuff. It feeds on decaying matter. Third one is Limnia luteola. I mean, Limnia luteola, this one is particularly very much invasive. This is a lot of aquarists have actually an axe to grind with this one because it is basically very invasive. It can spread through, if you take one plant from a pond and can accidentally drop it into another, or it can very easily proliferate. It will secrete mucus-like substance, lays eggs in there. Basically, it will stick to the back of the leaf and it will go on and on from there. Basically, very invasive. Same with the similar species, Limnia acuminata. Same proliferation, looks basically the same, and it will absolutely obliterate the aquascape if you accidentally introduce in your aquarium or small ecosystem, aquatic ecosystem. Next one is Endopnenorpis executus. Now, this species is from bivalve sections. You can see the difference between previous species and this one is has a sinistrate shell, that means a coiled shell. Gastropods basically, the first one, they carry their shell on the back of their, on their backs basically, and they travel. This one has a more laying down kind of existence. This next one is Lemididens marginalis. These come under the bivalve category of gastropods. These have 2 valves. The shell is made of calcium carbonate and it is attached with a very strong ligament. And they are basically filter feeders. They will take water from one orifice. They'll filter their whatever zooplankton or decaying material and expel the water which they have injected. That's how they basically feed. This one grows relatively large, about the palm of your hand. Piracy of evidence. This one relatively smaller, invasive as much. It is a indicator of pollution. If this one is present, water is relatively polluted. Corbicular stritella, it is also called as basket snail. It also a filter feeder, basically sedentary life. It has a relatively small number of neurons, or the nervous system is relatively less developed than the previous one, the gastropods. So this is Corbicular stritella. This is the reference. I mean, I identified this species from Handbook of Indian Freshwater Mollusks, Zoological Survey of India. Thank you everyone for listening to my presentation.
Question?
Hello.
Yes, sir.
Hello. I would like to ask one question. What is the ecological niche of this
What is the
mollusk?
ecological?
Niche?
Ecological. I,
Let her let her answer. Okay. Sir, let the candidate answer.
it is a benthic niche of the ecosystem.
Can you tell anyone means a chemical which is for which a particular mollusk is an indicator species?
I mean the basic chemical which is tested on this one.
No, any means toxicant.
So basically the fertilizers, liquid fertilizers, they are tested. I mean what is the effect of on liquid fertilizer or even pesticides. They are experimented on these mollusks, how it is, how the pesticide and the fertilizer affect their neural mass, the ganglia which they have present.
What is the importance of a mollusk in the food chain, food web?
Sir, mollusk, basically birds, migratory and local, they feeds on these mollusca and they help degrade the decaying matter as they consume it, secrete the excess, and that's why they are very good cleaners of the pond and provide relatively good food amount for the birds and small creatures or even crustaceans like crab and all.
Do they act as vector for helminth diseases in birds?
Yes, sir. The species Limnia luteola and the Limnia acuminata, they carry trematodes, which carries various diseases, and fasciolus diseases also get carried by that Limnia species.
Okay, thank you.
Fasciola hepatica, that parasite carries the fasciolus disease.
Okay.
Thank you, sir.
Thank you, Ms. Renuka. Now I call upon the next oral presenter, Mr. Jayendra Raut. Ms. Neha Badole. Ms. Prachi Mate. Ms. Jagruti Deshmukh. Mr. Aniruddha Deshpande.
Sir, should I start? Am I audible? Everyone without mic? Am I audible to everyone? Sir, am I audible to you? Okay. Am I audible now? Okay. So, honorable judges on the dais, respected teachers of the dais, my colleagues, my students, and my dear friends, myself Aniruddha Satish Deshpande, research student from Department of Botany, Government Vidarbha Institute of Science and Humanities, Amravati, and I am here to present the research paper on the topic sodium azide ethyl methane sulfonate and gamma rays induced high yielding and superior quality mutants in Linum ocedatissimum variety PKV NL260. So starting with the presentation, firstly introduction. As oilseed are one of the most efficient sources of different types of mineral nutrients in our daily diets, they are the sources of lipids, fatty acids, quality proteins, as well as then efficient energy reservoirs on which we can rely on. Okay, among those all oilseeds, flax occupies one of the significant position due to its unique fatty acid composition. Flax is rich in alpha-linolenic acid, which is almost 50% in its oil, and that's why it is very nutritional. It has several health benefits, like as an edible oil, as a functional food, as a fiber, as an animal feed, and as an industrial products. So, even though flaxseed is so much important in our diet, we can't use flaxseed oil in our normal diet because as it has highly unsaturated linolenic acid, and that's why what occurs, its oil get rapidly rancid. So that's why it can't be used for edible purpose. There are several earlier attempts in that case, like Natimoha and Roland. They have developed one of the low linolenic acid mutant of this flaxseed. Along with that, in case of Maharashtra specifically, Karpate and Chaudhary in 1997, and again Badere and Chaudhary in 2004, they have developed low linolenic acid mutant, but the yield is very low. That's why it is not beneficial for farmers to grow that crop. So if we consider the national scenario, Maharashtra is the least producer of that linseed, but if we consider the wider picture, that is worldwide production, India occupies 4th position. So according to Technology Mission of Oilseeds, if we want to reduce the import of these oilseeds from the foreign countries, we have to develop such cultivars and improve the oilseeds in order to reduce those imports from the foreign countries. So we have undertook this work. So material and methods. We have took this plant, that is Linum ucitatissimum variety PKV NL260. It is moderately yielding plant, which belongs to family Linaceae. It is moderately resistant to alternaria blight as well as powdery mildew and also to the bud fly. So we have took the germplasm from the Nagpur PKV. We have subjected it to certain doses of 2 chemical mutagens, sodium azide and ethyl methane sulfonate. And for irradiation treatment, we have sent the seed samples to the Bhabha Atomic Research Center, Mumbai. After that, the seeds were sown. Seeds were sown in order to raise the M1 generation. Threshing and harvesting was done. Next to that, all the seeds which were harvested in M1 population were again sown in order to raise the M2 population in 2 different fields. After that, the extensive biochemical analysis, morphological analysis, and recording of yield attributing traits, they have been done. And on the basis of desirable characters, the mutants were selected. They were again sown and carried out through the successive generation in order to check their stability and confirmation of characters. So these are results and discussion. So we have collected 4 most promising high yielding mutants which have about thrice or 4 times higher yield as compared to the control in case of their M2 generation. But in case of M3 generation, as it gets stabilized throughout the generations, we found the yield is reduced, but it is again 2 times higher as compared to the control in all the 4 mutants. These are again 2 other mutants. So there are total 4 high yielding mutants we have isolated from M2 population, and they are stabilized through the generation, and they are found to be true breeding. Next to that, bold-seeded mutants. The 2 most promising bold-seeded mutants were recorded. Like this one, this has twice the yield as compared to the control, and the test seed weight is higher as compared to the control. Another bold-seeded mutants, like this one, this is the BSM 13. This has one characteristic feature that the leaf size is considerably enlarged as compared to the control leaf size, and again it has also twice the yield as compared to the control in M3 and certain successive generations. Also, about 100 seeds, they weigh about 1 gram. So seed weight is considerably increased as compared to control. So we have carried out scanning electron microscopy analysis for that in order to check what are the reason behind increased yield in seeds, and we found that the surface cell arrangement in case of control and in case of this mutant, in the case of mutant, the angular diagonal distance is decreased as compared to the control, so cells are more compactly arranged, and that might be the reason which contributes its increased seed weight as compared to control. Next one is the dwarf with high yield mutant. This is one of the most promising mutant that I have isolated during my study. This mutant has twice the yield, and it has very lower height, about less than 45 centimeter. Usually dwarf mutants are very much preferred in case of linseed, because most of the linseed plants, they are generally have lodging problems in the field. That's why they are more susceptible to the fungal diseases, and as well as it lower down their yield. So these kind of dwarf mutants are always preferred because they usually do not get lodged in the case of plants, and also they can withstand the stormy and rainy winds. 3 high oil mutants are also isolated. These have considerably about 3 to 4% higher oil content as compared to the control, and yield is also considerably higher as compared to the yield of control. So there are about 4 low linolenic acid mutants are there. I am only giving here one. this linolenic acid mutant, it has lower linolenic acid content as compared to the control. Sorry. So in case of control, the linolenic acid content is quite higher, and that is the most probable reason, because it contributes to the rancidity of oil, and that's why oil can be used for the edible purpose. So we have developed one of the mutant among the low linolenic acid content mutant. It has lower linolenic acid content as compared to the control. So that can be used for edible purpose, and also yield is considerably, as you can see, it is almost equal to the control. Along with that, there are different mutants, like early flowering mutants are there, which are flower about 1 week earlier or 10 days earlier to the control. Then flower color mutations are also there. So while discussing that, if we consider the early flowering mutant, they have flowering period about 1 week earlier for the first flowering, and for the 50% flowering, they are about 10 days earlier to the control. If we consider the plant height, the lowest plant height was recorded in the dwarf mutant, that is about 39 centimeter. If we consider the capsule per plant, in almost all mutant capsule per plant are increased as compared to the control, with one or two exceptions. In case of these three mutants, the capsule number is quite high as compared to the control. If we consider 100 seed weight, that is the test weight, in case of both the bold seeded mutants, the seed test weight is considerably high as compared to the control. If we consider the yield, also in all the mutants about, yield is considerably higher than the control, with one or two exceptions are there. If we consider oil also, the same phenomena is going on. Lastly, the most important part, that is content of linolenic acid. In all the mutants, linolenic acid is considerably lowered as compared to the control. Control has the highest linolenic acid, that is 60.75%, while in all other mutant, linolenic acid is lowered, and least linolenic acid was found in low linolenic acid mutant. So conclusion, now here again we emphasize on the fact, which is the well-known fact, that induced mutations are very much more important and potent tool in order to improve the plant without alarming its original gene pool, as we done in a transgenic crop. So they have the wider acceptability in the social platforms also, and these all mutants, they might be useful in order to change the worldwide scenario as well as national scenario of linseed cultivation in our country and in a global level also. These are my references. Thank you.
How many different mutagens you have used?
Sir, 2 mutagens I have used personally, like sodium azide and ethyl methane sulfonate. We have given the treatment at our department.
Whether all of these are generalized or specific action?
I'm sorry, sir?
Whether their action is generalized or specific?
No, sir, they have generalized action actually, and that's why mutation are random. Means they cause point mutation.
And for how much generations you have tested the results?
Sir, I have tested for 3 generations, but now I have carried those mutants through successive generation. Now this year my M5 generation is going on. It is sown in Purna Nagar.
Okay.
In Amravati.
Anybody has any other question?
Hello.
Yes.
Sir, I would like to know if mutation has occurred in your experiment and increase in plant tissue, that particular plant tissue has occurred. Is it due to phytohormone effect? Is there any alteration due to mutation in phytohormones in the plant which has actually led to increase in the size of the leaf of the plant?
Yes, sir, definitely, sir.
Do you have any direct proof of it?
Sir,
or it is just a speculation?
No, sir, actually we always tested any kind of plant with respect to control. So if those variations are due to the environment, then in the same environment we are also taking the data of the control also. So if any kind of effect are found on the plant, it should be found on the control. If it shouldn't be found in the control, then that means it is somewhat new thing that we got. And for answer of this sake, I will just explain, like in case of linolenic acid content, like from oleic acid, this linoleic acid and linolenic acid were synthesized. In human, we don't have those hormones. So that's why we have to take those both beneficial omega-3, omega-6 fatty acid from the diet. Okay. So in that case, we might conclude that in which case that linolenic acid is reduced. At that time, that del 15 desaturase is there, one enzyme. Its activity might be reduced, due to which that fatty acid is reduced in that mutant. So that might be there.
आई वुड लाइक टू नो फाइटो हार्मोन्स पे डायरेक्ट है कि आपके पास एविडेंस कि फाइटो हार्मोन्स ऑल्टर होते हैं उसकी वजह से बड़ा साइज हो जाता लीफ का?
No, sir, I haven't worked on that direction.
ओके.
Thank you.
Thank you, sir. Now I call upon the next oral presenter, Mr. Mushtaq Dar, Mr. Mahesh Thengare, Mr. Nilesh Uparkar, Ms. Muskan Thakur and Ms. Nikita Katre.
Hello.
Good afternoon, everyone. First of all, I'd like to thank the organizers of this meeting for inviting us here today. It's an honor to address such a distinguished audience. First of all, I'd like to introduce myself. My name is Muskan Thakur and my co-presenter here is Ms. Nikita Katre. So the topic of our presentation today is bacterial quorum sensing inhibition, a new approach for drug discovery. So first, we need to clear two things. What is quorum sensing? And second, why do we need quorum sensing? So quorum sensing is basically that bacteria can communicate with each other. And bacteria can communicate with each other. It is basically a density-dependent gene expression phenomena that is shown by bacteria. We used to believe that bacteria are simple unicellular organisms for many years. But then when experiments were performed on Vibrio fischeri, we came to know that bacteria can actually communicate through each other through chemical messages. Okay, so this is how bacteria can communicate with each other. If you can see the red circles here, these are the autoinducers, which are the chemical complexes through which bacteria communicate. As mentioned, it is a density-dependent gene expression phenomenon. So these would only be expressed once the bacterial cell reaches a density, that is a threshold density. Now the second question, why do we need quorum sensing? So over here we have been using a lot of antibiotics. As we know that antibiotic kills only some amount of bacteria, but not all. This is because bacteria develop resistance against antibiotics. We need to overcome this problem, and the immediate solution to it is bacterial quorum sensing inhibition. This is because even if bacteria develop resistance against antibiotics, they would still not be able to infect an organism. This is the internal mechanism of quorum sensing. We have an I gene present in bacteria, which produces the autoinducers, which are the chemical complexes. Then we have an R gene, which is also known as the activator gene, which produces R protein. The R protein and AI, they form a complex together, and this complex goes upon the target gene. Once it goes upon the target gene, it's expressed phenotypically. Okay, this is the detailed explanation of how the autoinducers work. Now there are three types of autoinducers known to us. First is AHL, that is acyl homoserine lactones, which are the autoinducers present in gram-negative bacteria. Then we have autoinducer peptides, which are the autoinducers present in gram-positive bacteria. And third, we have autoinducer 2, which are the autoinducers present in both gram-negative and gram-positive bacteria. Now I'd like to handle the further presentation to Nikita Katre.
Thank you, Muskan. So as you can see here, these are various different processes which are under the control of quorum sensing. The first one is bioluminescence, virulence gene expression, sporulation, competence, swarming, pigment production, and at last biofilm formation. Now I would like everyone to look at this slide very carefully. These are some of the pictures of the processes I just mentioned. Like the first one is the picture of bioluminescence. The second one is that of swarming. See, you can see here how bacteria are migrating in groups using their flagellas. The third is that of virulence factor. And lastly, we have biofilm formation. So this was all about it. Now that we have talked about different processes in brief, I want to talk about the strategies we can apply so that there could be inhibition of quorum sensing processes. Now, if we talk about this process, that is inhibition of quorum sensing, it is pretty simple. Like either you can stop the formation of autoinducers, or if you are not able to do so, what you can do is you can use any enzyme to disintegrate that particular autoinducer. And at last, in worst scenario, if you are not even able to find any enzyme, what you can do? You can do the last thing, that is you can block the receptor side. Now this was pretty much about it. We'll move to the next slide. But before I proceed, I want to, if you remember, I talked about some processes, right? So out of all those processes, we'll be only talking about 3 main processes which are controlled by quorum sensing. The first one is the swarming, the second one is biofilm formation, and at last we have pigment formation. But before I go on that particular topic, I want to tell you that we performed many experiments. We use different chemicals, we use different substances to find if they have any potential to inhibit the process of quorum sensing. And what we found that tea extract, this was the thing which has at most potential to do so. So now getting back to the point, we'll start from swarming. But before starting this topic, I want to tell you what swarming is. So it is very simple. Swarming is the social behavior of bacteria where they migrate together over a surface, and to be more specific, in a semi-solid surface. So this is anti-swarming assay. Assay means a kind of experiment, we can say, or a laboratory test. So what we did is, we took an agar plate. You can see here, very clearly you can see here. And then we cultured the bacteria Pseudomonas aeruginosa. And we made 4 quadrants, 4 different quadrants, you can clearly see here. The first one, we kept it as negative. We didn't add anything in that, only the nutrient medium and the culture. Then the third quadrants were respectively A, B and C. In the 3 different quadrants, we added 3 different types of tea extracts. Then we incubated it, of course. After incubation, we got our results. And the results were, you can see, in A, B and C quadrants, you can see that the process of swarming was inhibited. But in the 4th quadrant, that is in the negative one, the process of swarming could still be seen because the tea extract was not added there, right? Now we'll move to the next slide and the next process, that is biofilm disruption assay. But before knowing about this, do you know what biofilm is? So I'll tell you. Biofilm means they are the glycocalyx materials which are secreted by bacteria themselves. And why do they do so? Just for their survival mechanism. So this biofilm disruption assay is another experiment we did. In this we took different test tubes, and in different test tubes we again added our nutrient media, we added our bacteria, the same bacteria. And at last we kept this first and second test tube different from others. Like in what way different? That in the first test tube we didn't add tea extract. In the second we only added methanol, and in rest of them we added different types of tea extracts. So, and again we incubated them. After that what we found is, you can see here in the first test tube, you can see a very prominent ring, and that ring shows the formation of biofilm. Then in the second test tube, we added only methanol. So you can still see the ring, but it is not that prominent, which means that that methanol was able to inhibit the process of quorum sensing, or what we can say is biofilm formation, but to a less extent. And in other test tubes you can see, in some of them no ring is formed, and in some the rings are faint, which clearly shows that tea extracts have the potential to inhibit the quorum sensing, but different types of tea extracts have different abilities, which is pretty much obvious. Now we'll move to the last process, that is pigment production, which is simple. That is the production of pigments. I'll not be talking too much about this because the kind of experiment is so much similar to that of anti-swarming. You can see these are the results of the inhibition of pigment production. Now, summing up our presentation, I want to say that we were successfully able to find a solution, that is tea extract, to successfully inhibit the process of quorum sensing and therefore decrease the chances of upcoming infections. And that was the end of our presentation. Unless anyone has anything else to say, that I think that's it. Thank you for coming here. Thank you for patiently listening to us. And I'm very gratefully thankful to our Professor Pradeep Hirapure sir for guiding us throughout our research. Thank you.
Thank you.
Now you may proceed with your questions if you have any.
Thank you. Next.
Next, Mr. Nayan Nandanwar. Mr. Ajay Dorlikar. Ms. Smita Gurde.
A very good afternoon, all the dignitaries on the dais, all the participants of this national conference. So today in this national conference, I'm going to present the research paper on morphodiversity in the genus Chlorophytum cargolar from Melghat Tiger Reserve of the Amravati district. Myself, Dr. Smita Gurde, Assistant Professor in Department of Botany, Arvind Babu Deshmukh Mahavidyalay, Bharsingi, Tahsil Narkhet, District Nagpur. So in the introduction, the Chlorophytum, if we say, the Chlorophytum is the very endemic species, very endangered species, and as here the farmer meets on this in this conference, so I want to say that most of the farmers take the yield of the Chlorophytum in their field, but they don't know the various species of the Chlorophytum. So I want to focus on that there are the various species of the Chlorophytum. Most of the farmers take the yield of the Chlorophytum borevillianum in their field. So it is basically a member of the Liliaceae family, and there are the more than 240 genera recorded. Overall with the world, there are the 4,000 species, and more than 200 species are present in the India. The distribution mainly in the tropical, subtropical regions of the world, and the native of the species is the South Africa, means 85% species of the Chlorophytum are present in the South Africa, and they are accidentally come into the India. So in Western Ghat, there are the 17 species are reported, and from Maharashtra there are the 11 species. And it is having the economic importance also, and it is very medicinally important also. Next. So the aims and objective of this work is to study the morphodiversity of the Chlorophytum, because there are the various species occur, and to evaluate why there is a morphodiversity in the species, what is the exact reason of the morphodiversity or speciation which leads to the evolution. So material methods, I collected the material, totally 4 species I have collected, Chlorophytum tuberosum, Chlorophytum laxum, Chlorophytum borevillianum, Chlorophytum arundinaceum from the Melghat Tiger Reserve of the Amravati district. Then I have studied about 70 to 72 characteristics, quantitative and qualitatively. And then why there is a so diversity, so I gone through the karyotype analysis. So these are the collection sites. So this is the morphological analysis, and you can see this is the habitat of the chlorophytum tuberosus. If there is a, means this species is found in the grass, we cannot identify unless and until there is a no inflorescence in the plant, because it look like a grass, because very, means this is very herbaceous species and we cannot identify this. So this is the plant, this inflorescence is there, stamens, pistil and fruit, and this is the identification made through the BSI. This is the chlorophytum laxum and various parts of this laxum species. This is again the grass-like structure. And whenever the inflorescence is coming out, then and then only we can identify this is the laxum. So you can see the differences in the inflorescence. But overall morphology is the same, and the main differences in the leaves and the tubers of the species. This is the chlorophytum arundinaceum. These are the differences in the flower. There are the 6 petals, but there is a differences in the morphology in their petals, tepals. Then tubers of the chlorophytum tuberosum, laxum, arundinaceum and boryvillianum. These are the seeds. And these are the 70 and 72 characters which I have studied for the morphodiversity. I skip here. So from this morphological analysis, we can say that there are the major differences found in the leaves, the tubers, and the inflorescences. And in the present study, the major differentiating characters also found in the filament of anther also. So why this is the morphodiversity present in the chlorophytum? So I study the cytologically, why there is a difference. So in the karyotypic analysis, I found that the C tuberosum, means chlorophytum tuberosum, is having the basic number X is equal to 8. Means it represents the 2n equals to 16 chromosomes. You can see in the ideogram and you can see the plates. There are the 16 chromosomes are present, and the red arrow indicate that there is a presence of this satellite. Means satellite, if there is a presence of the satellite, it indicates the asymmetrical karyotype, and asymmetrical karyotype leads to the evolution. This is the again chlorophytum laxum, that is a 2n equals to 16. Means in chlorophytum there are the 2 basic number, x equal to 7 and x equal to 8. So these 2 species represent the x equal to 8. And chlorophytum boryvillianum 2n equal to 28 chromosomes are there. Chlorophytum arundinaceum represent the 2n equals to 42 chromosomes are there, and these 2 species are represented x equal to 7. So this is the karyotypic analysis. And from the karyotypic analysis, I have to mention here that most of the asymmetrical characteristics found in the chlorophytum tuberosum and chlorophytum laxum also, and chlorophytum boryvillianum, because the satellites are present on their chromosomes. In the conclusion, as I earlier told that why I study the karyotype. So morphology in relation to the karyotype, we means there are, I also told that x equal to 7 and x equal to 8, 2 basic numbers are there, and x equal to 7 that represented by the chlorophytum boryvillianum and chlorophytum arundinaceum, and x equal to 8 represented by the chlorophytum tuberosum and chlorophytum laxum. So in the conclusion that there are the satellites are present, there is a morphology, again change in the chromosomes, that leads to the diversity in the genetical level, and that's why it leads to the plasticity in the morphology or phenotypic characters. Hence there is a morphodiversity occur in the chlorophytum. There are the references. Thank you very much.
Madam, is there any correlation between ideogram of 2 different species and morphological characteristic features of different species which you have studied?
Yes, sir. No, there is a correlation means there are the two basic numbers, and after karyotyping, then and then only we can say that this represent the basic means which species represent the x equal to 7, and which species represent the x equal to 8, sir.
You have studied about 72 different characteristic features.
Yes, sir.
So my question is that whether there is any significant difference in ideogram of these different species?
Yes, sir.
Is there
Yes.
significant difference?
Each and every species, totally I have studied 11 species, sir. Each and every species represent the different morphological characters in the chromosome also, and in the phenotypic characters also.
Okay. And from which part of plant you have studied these chromosomes? Only
Root tips?
only from root tip, vegetative part?
Yes. Only from root tips, sir.
Yes, okay.
Thank you.
मैडम एक क्वेश्चन था, अभी आप बोल रहे थे कि ये फील्ड में क्लोरोफाइटम के स्पीसीज जो ग्रो होते हैं, तो आपने अवेयरनेस क्रिएट की कि फार्मर्स को उसके इम्पोर्टेंस का?
सर एक्चुअली मैंने बताया नहीं, क्योंकि कैसे है सर, आई हैव गॉन थ्रू दिस साइटोलॉजी ओनली. इफ फार्मर्स को आपको बताना है तो उसको यानी ईल्ड भी देखनी पड़ेगी, क्योंकि क्लोरोफाइटम बोरीविलियनम इज अ हाई यील्डिंग स्पीसीज, बट आई एम नॉट श्योर दैट क्लोरोफाइटम लैक्सम, क्लोरोफाइटम ट्यूबरोसम और क्लोरोफाइटम अरुंडिनेशियम इज हाई यील्डिंग स्पीसीज और नॉट.
बट नेवरदलेस आपको अवेयरनेस क्रिएट करना चाहिए. बाद में यील्ड अगर कम ज़्यादा है,
यस डेफिनेटली,
और उसके मुताबिक कितना फ़ायदा लेना है फार्मर्स को,
यस
वो हो सकता है ना.
यस डेफिनेटली सर, आई विल डू दैट. थैंक यू.
Thank you, ma'am. I call upon the next oral presenter, Ms. Simran Kotwal.
Good afternoon, everyone. I am Simran Kotwal from Government Vidarbha Institute of Science and Humanities, Amravati. I am a PhD scholar. My topic is studies on genetic diversity for seed quality analysis with respect to mutation in rapeseed. I fits in category agriculture. Actually, today is the era of oil seed, because in 2020 and 2020, we have seen that there is 1,17,000 crore import of oil seeds from other countries. So I think that there is the continuous decline of oil seed in our country. For that purpose, I think we have to focus on oil seed production. So here in introduction, first I am comparing the profile of olive oil and rapeseed. There I have seen, you can see that the fat, that is saturated fat, that is 0.7 gram, that is almost very low than the olive oil. And in the second, you can see that there is given the brassica evolution in this portion. There is that. Am I audible clear, sir? Yes. Brassica rapa and Brassica oleracea and Brassica nigra is a diploid species, and Brassica napus and Brassica gentiana and Brassica carinata is a allotetrapoid species. And then next, there is given the comparison of dietary fats, where you can see that in rapeseed oil, the saturated fat have 7% among, that is very lowest than the amongst all the oils. And then you can see that omega-6 linolenic acid, that is 12%. And then omega-3 alpha linoleic acid, that is 18%. All these fatty acid composition is very high and low than the other oils. Then production wise, I think rapeseed mustards in India accounts 26%. Rajasthan is the major producer of rapeseed, and then among the leading producing countries, India at the 4th position. in historical findings, there are some important varieties that is released all over India, all over world. So the first variety that is released in world, that is in Canada. The variety is 00 type variety, and the name given to that variety is Tower. In my research work, I took 3 species. The first one is Brassica napus, the second one is Brassica gentiana, and the third one is Brassica campestris. What is the benefit and uses of Brassica, or you can say rapeseed? Actually, we are focusing on canola oil. So I am giving the uses of only that. You can see that it increases antioxidant because of the tocopherols, it decreases cancer cell growth, and there is so many. The main focus is to produce 100% non-genetically modified canola seeds by induced mutations. Material and method, I took 2 mutagens, physical and chemical mutagens. Then after that, the M1 seeds were grown in field. You can see that there is the image of vegetative phase. I took 2 generations in 1 year in different areas. Like if I took 1 generation in Amravati, Maharashtra, then the next generation I took in Jammu and Ladakh area. So these are the images from Jammu, Ladakh. You can see. Then you know that during the flowering stages, selfing and pollination is also important. These are all images from vegetative phase, flowering phase, and then the maturation phase. During the life cycle of the rapeseed, I have seen various disease and pest attacks. Then I did harvesting properly by taking all the necessary traits data. This process continuously we repeats up to M8 generation, and after that when we gain some characteristics trait that have high oil, high protein, and some bold seed mutants. So I think before giving them to farmers, we should detail protein analysis and oil analysis of that. So today I am focusing only on the protein and oil analysis, and some studies I did with scanning electron microscope. Okay, these are the accession mutants, you can say that, that is of different species like Bio 902, BCY2, and variety XL. These are the results, are electrophoretic pattern of seed storage protein, where you can see that the major protein we found in rapeseed, that is cruciferin protein. That means that if we found in our mutants, or you can say our variety, the cruciferin, that means that because the cruciferin majorly found in canola plants, so it means that our accession is on the way of becoming canola variety. These are during the protein analysis, generally I evaluated the low molecular weight bands. Among that, we have got 50 molecular weight bands. In that, I have also got some unique protein bands and some unique protein bands, and 7 monomorphic bands, and 46 bands or polymorphic bands. Then I did some cluster analysis by dendrogram, where you can see that in cluster 1, the Brassica gentiana, and cluster 2 have the major part of the accessions and Brassica species, and cluster 3 only includes 3321 mutants. The ancestor of 3321 is Brassica gentiana. Then I did some similarity coefficient matrix by Jaccard analysis. In that I seen that in Brassica napus the highest similarity value is 0.68, and in accession 4 to 7 the lowest similarity value is 0.26. These are seed scanning electron microscopic studies. What purpose of these studies is that if we have some mutants that have some characteristics feature. So we should know that who is the ancestor of that particular mutant. You can say that ki haan ye iska hai, but how? How can you prove that if you are not going by genetical way? So we can prove here that you can see that these are in figure A, this A, B, C, these are figure of Brassica jansea variety bio 902, and below all these figures are the accessions made by this control variety. You can see that there is no difference between these figures. Clearly you can see that there is only one type of pattern we have found. The pattern is a reticulate foveate type pattern. And then again Brassica campestris BCY2, in this same species we have seen that there is two types of patterns in accessions and variety. And in Brassica napus again we have got two types of seed coat patterns.
Ma'am, your time's up. Kindly wind up.
These are some data of one-way ANOVA analysis. Conclusion, I have found some stable accessions and the unique protein bands we have got in Brassica jansea, and I have seen 86% polymorphism in my study, and the diverse genetic diversity I have seen among them. Thank you. These are my references. Thank you.
For your study, how many samples you have considered?
Actually, sir, I took 3 species and 14 accessions, mutants.
And all these samples collected from which area?
Actually, the control varieties are took from all varieties generally are took from GVISH Amravati, and one Brassica jansea bio 902 are took from Punjab Rao Deshmukh Krishi Kendra.
Why you have not, taken trial of these varieties in Jammu, Ladakh and Maharashtra?
Yes, sir.
Why you have taken trial of these varieties in different localities?
Because before releasing any variety, we should have proof that actually these varieties are stable in different areas or not. Before releasing any varieties, it is very important to study their geographical analysis, and means it बहुत important है कि different environment में उनका analysis लिया जाए और देखा जाए कि वो वहाँ पे भी same हमको मतलब कि दे रहे हैं जो हमको product मिल रहा है.
Then you should also select another locality from South India also.
What, sir?
You have taken one from north, one from center, that is your location.
Yes, yes.
And why not other from South India?
Because, sir, actually I took two or three environment condition. One is warm area and another is cold area. So
Okay.
I think that is sufficient for study.
Okay, thank you.
Thank you, sir.
Thank you, ma'am. Next oral presenter, Ms. Kanchan Khapade. Okay, now I call upon M. Shravanan, his senior scientist from CICR.
Sir, shall I start, sir? Respected juries, delegates, students and friends, good afternoon to one and all. And this National Science Day Celebration 2023, I am presenting on conservation of land races, perennials and traditional varieties of Desi cotton collected from different agroecological zones, regions of India. Some of the important challenges we are facing, particularly global climate change and impacts. Nowadays we are seeing that particularly erratic monsoon rainfall, waterlogging and drought and other like deforestation, and nowadays increasing urbanization and desertifications. And so United Nations, they have set the goals like Sustainable Development Goals 17. It's endorsed by 139 countries, and 2030 we have to follow, meet these goals. Particularly 13 number, it's related with the climate action. And 14 and 15, that is related with biodiversity and conservation. These two are very important to this topic. And so the throughout the world, all the countries are gaining and awareness is created about plant genetic resources. So they are creating gene banks for particularly storing these seeds. If any war or natural disasters are happening, we can get the seeds from these different gene banks. Particularly this Svalbard Global Seed Wallet, it is present in the Norwegian country. It's very remote place, fully located in an ice area. Throughout all the countries, they have stored their seeds. If anything happens, we can get the seeds from this gene bank. And in India also, we have the National Seed Gene Bank that is in the NBPGR, that is National Bureau of Plant Genetic Resources. It is located in Delhi. And Government of India, they have developed so many institutions like National Biodiversity Authority, State Biodiversity Board, local biodiversity community for maintaining and promoting the conservation of seeds. And some of the organizations like Plant Variety Protection Authority and Farmers' Rights, they are also promoting farmers like seed protection awareness. So particularly I am coming to this cotton because every government is promoting different crop-wise based institutes. We are working on cotton. Cotton is important fiber crop, and basically it is a naturally perennial crop, but due to domestication it has become annual. Our India is the only country we are growing all the 4 cultivated species: Arboreum, Herbaceum, Barbadans and Hirsutum. And our country is the ancient home of the land races, particularly race of Bengalans, Cernum and Indicum of Arboreum, and Vaitianum of Herbaceum. And if you see the cotton statistics, India is first in area and production. But if you see in the productivity, we are 22nd position. Australia is the leading production wise. So see, 487 kg per hectare. Because some of the challenges like biotic and abiotic stresses leading this, pink bollworm, whitefly, this is the viral disease, that is the cotton leaf curl virus disease, and abiotic stress, that is waterlogging and drought. These are all important challenges in the cotton ecosystem. So why this happening? Because of narrow genetic base, because of domestication by our earlier farmers and modern plant breeding. Many of the alleles, see here in the wild species, there are so many alleles are present. But if we see the next, we have lost so many. And in the modern varieties, very less of alleles are present. So we have to get back to the wild ancestors. Then only we can get the crop. So we have to compare with the BT cotton, because introduction of BT cotton in 2002, later we have introduced the 2 gene products. So what happened? So the Desi cotton, particularly it reduced so much. Nowadays it's only 2% only we are growing. So we have to get back to the Desi cotton. We have to collect the indigenous cotton. So we can manage this, all the biotic and abiotic stress. And it is highly suitable for organic cotton cultivation, and some of the, you can see the tolerant to sucking pest, like many of the characters are best in the case of Desi varieties. And initiatives taken by ICR, we have taken literature survey and a letter written to Krishi Vigyan Kendra and forestry officials, and we collaborated with NBPGR, that is National Bureau of Plant Genetic Resources. We have started in 2011. Around 7 years we went for different exploration to different. So these are some of the collections we made. This is from West Godavari of Andhra, and this important land race. This is the Punduru cotton we have collected from Punduru districts, and some of the traditional knowledge. This is the fish bone they are using for removing small fibers, and these are the handmade equipments. And we have submitted application for Plant Genome Savior Award for Punduru. And this one, land race from, we have collected, this is Arboreum cotton, Karunganni cotton from Tuticorin of Tamil Nadu. And this is Uppam cotton, Herbaceum cotton from Tuticorin. And this is the, we collected one perennial important cotton from Raichur of Karnataka. And northeastern states, we have collected Cernum cotton from, these are all color cotton from Tripura area, and this is the traditional knowledge. And this is the Komila cotton from Meghalaya of Garo Hills. Here main is they are suitable for surgical and absorbent because the medical purpose it can be very much useful. And this is from Arunachal Pradesh. And this is the high altitude from Arunachal. And this is the traditional knowledge with the farmers of Arunachal Pradesh. And this one, Gujarat, we have collected Matio cotton from Gujarat. And this one, Wagad cotton from Matio, it's a herbaceum. And this is from Bandara from Maharashtra. And then we made morphological characterization based on flowers, all the dust characterization we have done. Then we studied the genetic diversity analysis. 3 groups are there. We have done the principal compound analysis and other analysis also. This one for arboreum, total 6 subclusters are there. Using Darwin software we have done. And this is the past status. Like many areas we used to grow all this desi cotton, but now we are growing only very less area, only 2% area in our country we are growing. And this is the status, only 2%. So only in the northeastern states and some of the Gujarat and very few packets only we are growing this one. So we have to collect all the cotton from through exploration, and we have to utilize in our breeding program. It's highly suitable for organic cotton cultivation. Thank you for organizer for giving this opportunity.
Thank you, sir. Thank you.
Thank you, sir.
Thank you, sir. Next, Mr. Samik Shlonare.
Good morning, everyone. Myself Samiksha Lonare. I am from MSc Botany from Institute of Science, Nagpur. I am here to represent my work on the topic studies on millet as a source of food and a value-added product. So nowadays, millets are termed as a staple food, and it has a very long history. Before 15 years ago, millets were the staple food, and now they are coming into the as a future crop in nowadays. Today we can celebrating this year as a 2023 as an international year of a millet and it is approved and declared by the government of India. Because millets have too many micro and micronutrient qualities like carbohydrate, protein, dietary fiber, lipid and vitamins, and they are too much health benefits for our health. As the population of India is increasing day by day, so the availability of the food resource is limited to feed our overpopulated mouth. India is home to more than 1/3 of the world malnourished children. By contrast, and the country had also become the hub of health disease after the pandemic. So the country has put itself under a double burden of a malnutrition. Indian millet are nutritionally superior to wheat and rice as they are rich in protein, vitamins and minerals. Millets are the group of small round whole grain from the family Poaceae, and they are distinguished into the 2 kinds of millet. Some large millet and the small millet. Large millet consists of bajri, tangni, jowar, ragi, and small millet consists of sawa, kutki, korle, and white and black panino.
In spite of explaining these things, would you come to please your plates, figures,
Okay,
table,
sir.
if any, and try to conclude it in a short time.
Okay. In this work, I just visited to the 10 supermarket and interviewed with the people, local people especially, and visited 2 exhibitions for the collection of the images and the types of a millet. During the survey, I found some bakery product, flakes and pops, extruded product, fermented products, and these are the various type of a millet, types of millets, and their value-added products that are available in the market. These are some of the products like cookies, etc. And these are the 12 types of a millet. Their botanical name, vernacular name, area of cultivation, place of origin, and their biological activity. As they are nutrient, consist of much of a nutrient, they are health benefits. This is the map of India showing where the millets are cultivated in most of the amount. So as a result, I got to know that local people, especially in the urban areas or especially among the teenager, they don't know much about the millet. As I am a teenager, being a teenager, I also don't know about the millets. And it is very essential for the people to know the more value, nutrition value and health benefit of the millet, as many of us are diet conscious, and so we can consume more of the millet instead of eating junk foods and other foods. These are some references, and thank you.
Thank you.
Oh.
Here we
Is
end
there
up
any
the-
question from audience? Just one question only. Okay, thank you.
Thank you, sir.
Thank you.
Thank you. Here we end up the oral presentations of life sciences. From physical, chemical and other sciences, I call upon the next oral presenter, Rajshekhar Okre. Mr. Rahul Kurzekar, Dr. Nand Kishore Padole, Ms. Uttara Tagde, Mr. Praveen Dhakite. Yes, sir.
So thank you all the judges for your valuable time for judging all the presentations. Now I request Dr. Archana Mishra madam to handle the further proceeding of the valedictory function of this national conference and farmer mates. Thank you. Now we are starting valedictory function with the guest of honor, chief guest, and back student, please come forward. Please sit in front. Shortly we are going to start valedictory function of this national conference. I request Honorable Dr. Pramod Ramteke sir, ex-director research and Dean Allahabad Agriculture University Prayagraj, Uttar Pradesh, India, to come on the dais. I also request Honorable Dr. Praveena Khobragade madam, Principal Dr. Ambedkar, Dr. Baba Saheb Ambedkar College of Law, Rashtrasant Tukloji Maharaj, Nagpur University, Nagpur. I also request my principal, Honorable Dr. B.A. Mehere madam, to please occupy the seat. I request Dr. Chinna Babu Naik sir, Senior Scientist CICR, and Dr. J.H. Meshram, Principal Scientist ICR CICR, to please come on the dais. है कि नहीं, सबको मिलना चाहिए ना,
The chief guest for this valedictory function is Professor Dr. Mohan Khedkar, Professor in Electric Engineering Department, Vishwasharaya Institute of Technology, VNIT Nagpur, and ex-Vice Chancellor Sant Gadge Baba Amravati University, Amravati. So I request all the participants to welcome the ex-Vice Chancellor Sant Gadge Baba Amravati University, Amravati, Honorable Dr. Mohan Khedkar sir.
A warm good evening to everyone. Welcome, sir. Please have a seat. A very good evening. Dear friends and students, today you have enjoyed the National Conference on Innovations in Science Technology for Future Sustainability and Farmers Meet on the Agri-Innovations for Sustainable Productivity. I am sure this must have been a wonderful experience of your life. The basic concept of lab to land. Science Day, we celebrate it on every 28th of February. But what is the use of science and the technology if we cannot provide it to the very downtrodden people of this country? And I must say that yes, here we have a faculty like Dr. Subhash Somkuar sir, who took all the efforts to make this program and this farmers meet a success and take all the concepts of biology or science from this very laboratory to the land of the very own farmers of this country. I congratulate Dr. Subhash Somkuar and Principal of Dr. Ambedkar College, Dr. Mrs. B.A. Mehre Madam, for taking such initiative step. Today, to mark this untired efforts of the whole team, we have gathered here for this valedictory function. I request the dignitaries of the dais to please do the honors of felicitating and paying homage to the scientist of all the eras. As I always say, that Buddhism was an era where it was actually science was taken from lab to land. Buddha, the greatest scientist, made sure that through his preachings, he enlightened and awakened the mind of every person so that his concepts were followed by. I request our guests to please pay homage by offering flowers to Tathagat Bhagwan Gautam Buddha. And dear friends, on this very pious land of Deekshabhumi, Dr. Baba Saheb Ambedkar made sure that whatever scientific thinking Buddha had given us, he made sure that he writes it into the constitution and gives it back to you so that you can take the privilege and do all the necessary things which you have as a basic right. So these two stalwarts of humanity are really the persons who have changed our minds. Lighting the candle is simply a way of Buddha saying Atta Deepa Bhava. You yourself are so enlightened that you do not need any external force or the external energy to bring up that light from you. It is within you. Only thing is that you have to recognize it and bring it back.
Thank you so much. Moving ahead, it is a time to respect and say thanks to the dignitaries on the dais for their elite presence. I request the very dynamic yet friendly very strict yet humble, very much intelligent yet ground to earth, none other than our principal, Dr. Mrs. B.A. Mere, madam. I request her to please welcome Professor Dr. Mohan Khedkar sir, ex-Vice Chancellor from Sant Gadge Baba, Amravati University, the very own place, my very own land. It gives me immense pleasure to welcome you, sir, on this Deekshabhumi and Dr. Ambedkar College. We are really blessed to have you today as the president of this function. Welcome, sir. Thank you so much. Madam, you deserve it. I now call upon the very much energetic, intelligent Dr. Subhash Somkuwar sir to please welcome Dr. Praveena Khobragade madam, Principal Dr. Babasaheb Ambedkar College of Law, RTMNU, Nagpur. Madam, it is really nice to have you here today, and it will be our pleasure to hear you. We are just awaiting for that moment. I call upon Dr. G.H. Meshram sir to welcome Dr. Pramod W. Ramteke sir, ex-Director, Research and Dean, Allahabad Agriculture University, Prayagraj, UP. Dr. G.H. Meshram sir. Please welcome Dr. Pramod Wankhede sir. Wankhede sir is also a faculty. Ramteke sir. Dr. Pramod Ramteke sir is also a faculty at our college, and I must say that the people like Dr. Ramteke sir, who are ex-director and dean, still they are so humble and polite. Thank you so much, sir, for gracing the occasion. I request Dr. G.H. Meshram sir to please stand there for a moment, because it is a time to welcome the biggest pillar of this institution, Dr. Mrs. B.A. Mere madam, the principal. Guys, I must say, students, a very big round of applause, because she is the person who gives you access. I must say, madam, that in the history of Dr. Ambedkar College, you are going to be the most popular principal, because the students never have to seek permission to enter your cabin, and that is what the administrator is all about. Thank you so much, ma'am, for being there. I request Dr. Mrs. Aarti Vazalwar to welcome Dr. Subhash Somkuwar. Friends, had it not been the efforts of Dr. Subhash Somkuwar, we wouldn't have been standing here today. It is the basic idea of this man. And he does not only have those ideas, he makes sure he brings those ideas into a reality and turns them into the fruitful events like this. Heartiest congratulations, sir, for the successful completion of this program. I request Dr. V. Chinna Babu Naik, sir, to please welcome Dr. G.H. Meshram, sir, Principal Scientist, CICR. I must say, CICR has always been there with Dr. Ambedkar College, be it funds, be it their research attitudes, and be it with their research supports. Thank you so much Team CICR for being there, and thank you so much Principal Scientist Sir Dr. G.H. Meshram Sir for being there. Thank you. I request Dr. Rahul Kamble sir to please welcome Dr. Mrs. Aarti Vazalwar, co-convener of this program and an HOD of Physics Department. I must say Aarti is also a very enthusiastic person and takes lots of efforts to arrange such activities. Welcome Aarti. And last but not the least, I would call Dr. V. Chinna Babu Naik sir to welcome the ever supporting, intelligent, a person who works in silence. His action speaks more than words. What do I say for Rahul Kamble? He is the man. And I must say your popularity is growing more, Rahul sir, and that is a good sign. Guys, he never thinks that he is not holding any government or an official position, and still he is giving his untired and effortless contribution to the Department of Botany. Dr. Rahul Kamble and Department of Botany these days are an inseparable team, and not only Department of Botany, Dr. Ambedkar College as well. Dr. Rahul Kamble has put tremendous efforts in this 13 years. That is what I have seen. Thank you so much, Dr. Rahul, for being there. I request Rahul Kamble sir to please welcome Dr. V. Chinna Babu Naik sir. Yes, sir, थोड़ा सा exchange कर लिया है हमने, बाकी कुछ नहीं. I now call upon Dr. Subhash Somkuar sir for introducing our today's renowned speaker, Dr. Pramod W. Ramteke sir. He is an ex-director of research and dean, Allahabad Agricultural University, Prayagraj, UP. Over to you, Dr. Subhash Somkuar.
I very briefly introduce before you Professor Dr. Pramod Ramteke. He is currently adjunct professor at Department of Life Sciences, Mysore University, Mysore. He has 36 years of teaching and research experience and contributed significantly towards developments of microbe-based eco-friendly processes to the sustainable crop production and utilization of agriculture wastes for production of biofuel and industrially important enzymes. He handled 13 minor and 15 major research projects sponsored by UGC, DST, and so many international sponsoring agencies. Because of his dedications to the research and teaching profession, he is recognized as a fellow of various national and international institutions and academics. He is elected fellow of National Academy of Agriculture Sciences, Biotech Research Society of India, Royal Society of Biology, Academy of Microbial Sciences, National Academy of Biological Sciences, Mycological Society of India, and International Society of Environmental Botanists. In view of his outstanding contributions, various awards and honors were conferred on him, like BHU Centenary Awards, Excellence in Science and Technology Awards, J.C. Bose Gold Medal Awards, Dr. J.C. Edward Medal Awards, Professor K.S., and so many awards he has bagged in his academic careers. He mentored 39 PhD scholars and 7 postdoctoral fellow students and 4 patents, 2 technologies, 5 databases, and 323 gene sequences in National Chemical Bioinformatics USA to his credit. He authored, edited 16 books published by Spengers, elsewhere, Ville, Taylor and Francis, and so on, including Pliers, ACS, and whatnot. He has 6,380 citation index as per the Google Scholar agency. He is nominated as a subject expert and chairman state and national level environmentals and animal ethical committees by Ministry of Environmental Science and Climate Change, Department of Science and Technology, Indian Council of Agriculture Research, Indian Council of Medical Research, Council of Scientific and Industrial Research, Government of India. He was visiting scientist to Hungary, USA, UK, Belgium, Turkey, Czech Republic, South Korea, and so many other countries. He is a member of 18th Indian Scientific Expedition to the Antarctica. With these brief introductions, now I request Honorable Dr. Pramod Ramteke sir to please guide this August gathering. Dr. Pramod Ramteke sir. Thank you.
My deep respect to proponent of peace, Lord Gautam Buddha, and Vishwaratna Dr. Baba Saheb Ambedkar, all the dignitaries on the dais and off the dais. We have less time. And this very important conference, we have all gathered together. And this conference have two important components. One is innovation and another is sustainability. And we all now know that we are facing a problem of sustainability. So much environment we have already damaged. And now it's a time to restore it back. In this context, three important components are there. Okay? You have a people or society, then you have a environment, and then you have a economy. These all three important components composes actually directly related to the sustainability. Now, to fulfilling or to restore the sustainability, we have been doing so much, okay? But still there is a need for innovation. And innovation is not solving problem in old ways, okay? There has to be an element of new thinking. Then only this could be possible. So, if we talk about sustainable, it's not only doing the, not only harming, okay, harming less, but also doing good more. So we have to start off in this line. Maybe I don't want to take so much time with when we think of when we deal with the environment. I think I want to sum up with this thing. अगर फुर्सत मिले पानी की तहरीर को पढ़ लेना, हर एक दरिया हजारों साल का अफसाना लिखता है. So this is a very, very important, this thing, that when we think of using anything, we should think of the past several thousand years of history. I take this occasion also to thank all the organizers, especially the Ambedkar College and also Central Cotton Research Institute for bestowing me or honoring me with the first Professor Meghnad Saha Lifetime Achievement Award. Thank you very much.
थैंक यू सर, दैट वाज इंडीड वेरी इंस्पायरिंग. और जब दरिया की बात आपने कर ही दी है तो मैं बताना चाहती हूँ कि अंबेडकर कॉलेज का हर एक बच्चा दरिया है. ये आपके लिए है स्टूडेंट्स. हम दरिया है, अपना हुनर जानते हैं, जहाँ से भी गुजर जाएंगे अपना रास्ता खुद बनाएंगे. So with these words, now moving forward, I would like to announce the Young Scientist Award of this conference, ISTFS 2023, the Young Scientist Award 2023
It goes to recognize outstanding research in green technology development and design for sustainable future to Dr. Ganesh C. Nikharje, Assistant Professor Botany, R.K. Talreja College, Ulhasnagar, India, and Postdoctorate Fellow of Jeonsang National University, South Korea. A big round of applause for him. He is not here, but sir, of course, thank you so much and congratulations for this Young Scientist Award 2023. I hope the organizers will make provisions to give the award to them, whatever ways they have decided to. The next felicitation is of the judges for the oral presentation. I request Khirkar sir to please do the honors and felicitate Dr. Zaki from Institute of Science, Nagpur, for being a judge for the oral presentation sessions today. Welcome sir, and thank you for helping us out with the judgments. Our next judge for today's oral presentation is Dr. Dadure sir. Thank you sir for helping us out with the judgments and doing the tedious job. Thank you. I also call upon Dr. Rohan Thavre. Sir, the next felicitation is of Dr. Rohan Thavre from our very own Department of Biochemistry. Dr. Rohan Thavre, another young enthusiast who we have here and a very research-oriented person. Thank you, Dr. Rohan Thavre, for always being there. Yes. Dr. Nagrade, I please request you to please be present on the stage. Dr. Saxena sir. Dr. Nagrade sir, followed by Dr. Saxena sir. Thank you so much. Dr. Saxena sir. Thank you sir. खेड़कर सर मैं आपको थोड़ी सी और तकलीफ देना चाहूँगी. You have to also encourage budding Buddhas and young scientists, that is our dear students who have won the prizes for best oral presentations. Before that, I request you to please welcome, Velmogan sir. He is the co-chairperson of this national conference, and for the oral presentation, I please request Velmogan sir to please be here. Sir is here? Sir, nahi hai. Sir. Chinna Babu sir is going to take on his behalf. Beta ye de. Sir, beta. बोल वैसी ही चली गई. वो बच्ची वैसी ही चली गई. हाँ वो सर्टिफिकेट है ये देना है. अरे तो सर्टिफिकेट मेरे हाथ में ये दोनों क्या कर रहे हो आप लोगों को कोऑर्डिनेशन नहीं है मैं नाम लिख रही हूँ डोंट डू दैट Now is the time for the oral presentation awards. Third prize goes to Swanand M, CICR. Third prize. Sarvanam, sorry. M Sarvanam, CICR. And Anisha Rahate from Dr. Ambedkar College. They have won the third prizes. Anisha Rahate, please come. Sarvanan M. Sir from CSCR, congratulations. Anisha has also won the third prize and she belongs to Dr. Ambedkar College. Second prize goes to Avinash Aniruddha Deshpande. He's from Amravati. Avinash Deshpande from Amravati. He has bagged the second prize. I know. It's Aniruddha Deshpande. Rakesh Dharwad from Karnataka. Mr. Rakesh Dharwad has won the second prize. Congratulations Aniruddha and congratulations Rakesh. And the most awaited, that is the first prize goes to Dr. Lal Singh from Akola. Dr. Lal Singh, anybody who's going to take on his behalf? And Dr. Garpaile from Tumsar. On behalf of Lal Singh, congratulations for winning the first prize and Dr. Garpaile. Dr. Garpaile from Tumsar. Heartiest congratulations to all the winners. Dear students, as the consolation prize for this oral presentation goes to Chandrasekhar Khare from Champa, Krishi Vigyan Kendra, Jangir, Champa. Raipur, Chhattisgarh, Chandrasekhar Khare. Congratulations, sir. Dear students, as we have already started late and we are running late and out of time, and our ex-Vice Chancellor sir has to move on with some pre-engagements. So I request Dr. Mrs. Aarti Vazalwar to please give a brief intro of Professor Dr. Mohan Khedkar and welcome him for his presidential address.
Thank you, Archana. It is both my honor and privilege to be able to introduce Professor M.K. Khedkar. He was the ex-Vice Chancellor of Sant Gadge Baba Amravati University, Amravati. He was twice head of Electrical Engineering Department at VNIT Nagpur. He was conferred the honorary colonel by NCC Directorate, Maharashtra, Mumbai in 2012. He was also conferred honorary fellowship by ISTE for the year 2011. He received the Maharashtra State Best Engineering Teacher Award for the year 2006. He was the ex-dean academic and ex-dean administration VNIT Nagpur. 6 students till date have completed PhD under his guidance. He He has published more than 150 papers at national, international level, journals and conferences. These are only some of the features, friends, of his extensive biodata that I could just spell out. So after his brief introduction, may I request Mr. Mohan Khedkar to kindly address this august gathering.
A very fine good evening to all of you. The dignitaries sitting on the dais, the participants, the judges, the professors, and my dear students. Normally, in a valedictory function, if you have to speak at the end, that's the most difficult thing. You know, in Bombay, there was one deputy mayor who was very popular. His only condition used to be that he will talk at last. And you know, when he used to come to the dais, what he used to say? I agree with the views of previous speakers, thank you. And so he became very popular. Unfortunately, being ex-vice chancellor, I can't say so. And so you have to bear with me for some time at least. And the conference which is there, which was held right from the morning, Innovations in S&T for Future Sustainability, wherein they said that they were having, I don't know the exact number, but participants. Here I can see only the judges and the students mostly. Maybe that they may be busy with something else. Or probably, you know, in many cases how the conferences used to occur. Being a vice chancellor, I used to have so many conferences. And when it is a one-day conference, you know, people start pouring in till lunch. They finish up the lunch. Post-lunch, it's very difficult to hold the audience, as it is at the end. And then start leaving. Except for probably if there are certificates which they have to show either to their institute or to their university. In that case, they have to be present. And I thought that at least the prizes or the judge's certificate will be given later, so that when I speak, at least somebody should be there in front of me. Because this is compulsory, so they can't leave. Judges hopefully will not. And I heard that the Ambedkar students are very good, so hopefully they will also manage. They have started. See, जैसा मैंने बोला उठ के चले गए वो. So that I know well. Though the conference also they said that they are having the farmers meeting too in the morning. Of course, I will talk about the innovations in S&T. And then we'll speak about only the words of it, and probably I think that will consume enough time for me. When we talk of sustainability, what we mean by sustainability? Sustainability is something, some innovation, which you give to the world today so that it should not reciprocate back neither to you, neither to your children, to grandchildren and great-grandchildren. We know you use a battery, we just throw it. We use a plastic bottle, we throw it. And then we think of a sustainable environment. Because you know, as a human being, I am happy because, oh, everything is available to me. Why I should worry about what will happen over a period of 50 years or 100 years? Who am I? I don't have to worry. The global warming is there. All the time we talk about everything green. Green, 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 you must have heard of so many times. Why so? Why we need it? It is for the sustainability. And when you ask me what is needed for sustainability, I will say few things. These are my own, you may or may not agree. The basic things which are required are roti, kapda or makan, the food, cloth and shelter. That was the requirement few years ago, maybe during independence or pre-independence. To that, over a period of time, I will add education and the primary health center. And now, if I talk of 2023, probably in that I can also add internet. Internet is तो कुछ कर ही नहीं सकते ना. So probably that also may be needed. And so with these 6 things, we can think of some sustainability. The another word which was there was the R&D. That is a very dangerous word, I will say. The research and development. So many papers come up, right? We check the plagiarism, we have citations, we have indices, so many things to judge it. How many of them get converted into practice from lab to land? How many innovations they really go? That's a simple question which you can ask to yourself. I'm not going to answer. I will keep few questions for you. Or is it simply for getting my degree, getting me a job, or getting promotions? If that is there, fine. Nothing wrong. The other thing is, let us say you develop something, at what cost you have developed it? Is it really affordable? You know, the artificial leg was developed, what we call it as a Jaipur leg. Jaipur foot, they say. And then they used to say that by wearing this foot, you can play even football. Fine. That's very nice. But what is the cost? The cost is 20 lakh rupees. Is it affordable to the Indian? Why Indian? Is it affordable across the world? So when we are doing research, we have to also see to it that it can reach to my people, my rural people. When we talk of India, normally when I talk, I always say India and Bharat. India is what you see, all these malls, airports, metros. And the Bharat is probably the farmers which you might have seen in the morning. That is Bharat. But then it doesn't mean that only in India R&D can happen, only in the institutions it can happen. No, sorry. It even happens in Bharat. And probably that is the innovation, the invention which they do. They are need-based, they are location-based. If they are in and around the region, probably they will find out something which is most useful to them. And in a rural India, why in rural, even in urban India, there is a very common term which we use in India, जुगाड़. जुगाड़ टेक्नोलॉजी. सर ने और उसको बढ़ा दिया. सर करते हैं लगता है. इतना पता है उनको सब. करना ही पड़ता है बोलते. मैडम ने भी एंडोर्स कर दिया अभी. हाँ हाँ, वो ऐसा है ना, पहले लड़के को पूछा कि शिव धनुष्य किसने तोड़ा, तो लड़का बोलता मेरे को नहीं मालूम, मैंने नहीं तोड़ा. टीचर को पूछते, सर मैंने नहीं तोड़ा. प्रिंसिपल के पास जाते, प्रिंसिपल तो मैंने नहीं तोड़ा, मेरे को नहीं मालूम. Now, you know what is happening? That jugaad which we are talking of is really useful. If you go to any rural area, Bhamragad, and you will find that the Adivasis, what they have invented, we won't recognize them. That is a need-based invention. That is the invention which is needed for that place, and at a reasonable cost, at an affordable cost, most of the times it is free of cost. Whatever material is available, they have made out of it. And probably instead of my PhD, probably that innovation is much better. The problem is we are unable to recognize them. So when we talk of R&D, we have to also see to it that whether it is reaching to a poorest man from the remotest corner of my country. If you ask me then as to what is in my mind as an electrical engineer, what the innovation could be, what I can do, what I can contribute. Because no, always when it comes to ये करना है, वो करना है, क्या बोलते हैं हम? अरे इतने तो लोग हैं. There are so many faculties, somebody will do. I will not. Is it not? So many students, one student will do. Online exam था, बाकी सब WhatsApp पे डाल देंगे. I will not. I will just tell you, it is you who will have to start. Who is that somebody? Why I should wait for somebody else to start? I can give my own example. When I say so, I mean it. Innovations are not new to us. If you open any Sanskrit shloka, there is a book, Viman Shastram, and for your information, I would like to tell you, it was not in 1905 when Wright brothers invented the first aeroplane. It was in 1895. One man, Mr. Tarpade from Mumbai, the aeroplane was there on Bombay Chowpatty, Girgaon, those who are acquainted with Bombay. It flew for 15 minutes, which was being witnessed by the mayor of Bombay, the governor, and then the Maharaja from Baroda. And he developed that aeroplane based on the book Viman Shastram. Simply, you know, in India, as it happens again, that whether it is a turmeric, haldi, whether it is anything else, we never go for any patents. We never know that this is innovation. We just do it. And so it's time that if you do the innovation, and which is really the area in which you live, you do it. Right? I think अभी तो समय हो गया लगता है और एक लोग जा रहे हैं. फोन कर रहे हैं. कोई बॉयफ्रेंड का फोन आया लगता है. इतना अर्जेंट है ना? पेरेंट्स का है? ओके ओके. नहीं, इतना अर्जेंट फोन मेरे को नहीं आता है ना, इसलिए. और मेरे को आया तो सीधा घर में ही जाना पड़ता है फिर. दूसरा चॉइस ही नहीं रहता ना. I think those who are married, they will understand. Others may not. Students may not understand right now. Okay, so that was about the R&D and sustainability. So as I said that already it is available. Everything is available with us. Even if we decode that. And so I started a subject, Sanskrit for Technical Development in VNIT. The Sanskrit is an art subject. Technology is a technical subject. When I started to write a book, I couldn't get a publisher. I had to take some local publisher. So what I mean to say that everything is available with us. Our brain, all the US, the dollar, the international currency, the world's leading nation, is run by whom? They are all Indians. If you go to Texas state, 75% of them are Indians. You go to Google, you name the industry, and you will find Indians. Now the question is that if they can do that well in America, why they are unable to do it in India? The environment which we want, I think you know the word environment. That has come from the French word environ. Right? Environ means surroundings. And when we talk of sustainability, we talk of environment, we talk of the surroundings. Surrounding means maybe a human being, maybe an animal, maybe this building, maybe anything else. Everything becomes surrounding. And so the innovations which I thought of is the hydrogen. What I imagine tomorrow means not exactly tomorrow, maybe 10 years down the line. You know what is our aim? We want net zero by 2070. I think you know the concept of net zero. The carbon net zero, that is the carbon emission will be zero by 2070. Our CO2 emission, when we measure, per capita CO2 emission in India is less. We may say that the pollution is more. We know why it is so? Per capita, so whatever is the total CO2 produced divided by the total population. So a larger population is always not a problem. At times it gives some advantage also. The denominator being more, the per capita CO2 emission becomes less. And so net zero means you want that to be zero. And that is what is sustainability. Now how to achieve it? As I said that, I will think of maybe 10 years, 20 years down the line, as you are having electricity line right from Nagpur to Bombay, I am expecting a hydrogen pipeline from Nagpur to Bombay. Maybe through Samruddhi Marg, maybe some other road. Now you may be asking, what is that? Why you want hydrogen? What is it? You see, as soon as you are having a hydrogen, you can tap it at any place, no, Amravati, Akola, Nasik and Shirdi, wherever you want. Why it is? Hydrogen, you can make CH4, methane, you can use it for heating. You can use the hydrogen vehicle. Our honorable minister, transport minister uses a hydrogen car. You can also use it for producing electricity with the help of a fuel cell. And the height of it is, it doesn't produce any pollution. So it will be a sustainable solution. So something should come to your mind also that this is what it should be. This is how, if each of us think, a day is not far off when we can really achieve a sustainable development. Probably I think most of them are biology students, they might be knowing algae. Algae, you know, it absorbs CO2, it can give out the petroleum products, and again, you will find that the algae is formed because of the sunlight. So in a closed loop it works, and it will have all the advantages only. So we have to think of some such solutions. If we can get these solutions, probably, instead of calling ourselves as a developing country all the time, when the time will come, when we will say that ours is a developed country. All the time we are saying developing, developing, developing. Till what? After 75 years of independence, still developing? When will be developed? And when I say developed, it is only you and me, not somebody else. Remember one thing. I am again pinpointing that thing. कोई तो भी करेगा ऐसा नहीं है. एक अकबर बिरबल की कहानी बता के खत्म करता हूं. अकबर पूछता है बिरबल को, भाई यहाँ के लोग कैसे हैं? तो अकबर क्या बोलता है, अरे यहाँ के लोग तो मेरे लिए जान दे सकते हैं, अपना खून दे सकते हैं। बीरबल क्या बोलता है, साहब खून वगैरह तो जान दो, एक गिलास दूध दिया आपके लिए तो मैं समझूंगा कि सब लोग आपके हैं। सो व्हाट दे डू, अमावस का दिन लेते हैं रात को, बायोमेट्रिक तो नहीं था, अंगूठे लगाना पड़ता था, आजकल बायोमेट्रिक आ गया। तो बोले हर एक आदमी जो है वो गांव का, लेट अस से देयर आर 1000 पीपल, एक आदमी आएगा रात को अंगूठा लगाएगा, पहचान में नहीं आना चाहिए ना कौन आया कौन नहीं, और एक गिलास दूध वो टंकी में डालेगा। रात भर सिलसिला चालू रहता है। एंड अकबर वॉज वेरी हैप्पी, ही वॉज टेलिंग बीरबल ऑल द टाइम, देख मैंने बताया था सब लोग आएंगे, सब लोग आएंगे, देख 1000 लोग आ गए, 1000 अंगूठे है, बायोमेट्रिक हो गया। जैसा आजकल होता है ना, एक बार सुबह मार दिया बायोमेट्रिक 10 बजे, एक बार 5 बजे, दैट्स ऑल। बीरबल सेज वेट फॉर द मॉर्निंग। सुबह क्या दिखता है वहां पे मालूम है? इट इज ऑल वाटर, देयर इज नो स्टिंच ऑफ मिल्क ऑल्सो। व्हाई इट हैज हैपेंड? एवरीबडी थिंक्स, हर एक ने क्या सोचा, जो 999 लोग हैं वो तो दूध ही डालने वाले हैं, मेरा एक गिलास पानी जम जाएगा उसमें। ऐसा करके सभी 1000 लोगों ने पानी डाला था उसमें, क्योंकि मैं नहीं करूंगा ना, somebody else. So again I am telling you, it is you, especially for the students I am telling, the energy which you have, they all said that they are all from the biology background and botany and all that. You know what is the energy stored in a seed, right? When you go roadside, and if you see those mountains, you will find that from that seed, the tree has grown, which has broken all that great stone. So that's what is the energy with you. So simply be positive, change your attitude and aptitude, and you will find that you can win the race. You can get the success in life. There is nothing like a shortcut for achieving a success. So you do R&D, you do it for sustainable development, right? So I think with that, I again thank the organizers for calling me over here. And I again thank you, because every speech has a happy ending, because everybody is happy when it ends. Thank you.
Thank you so much, sir. What a wonderful way of expressing the views and giving examples of sustainability. Sir has rightly mentioned that India has a rich heritage of science, and for that we do not need any proof. Nalanda University itself was the biggest proof what Buddha had given to us. Now it is up to us, when Buddha says mind is everything, it is up to us what to take and what to leave. He always says that be the awakened one, and that awakening, my dear friends, is going to come only when you follow the path of Buddhism and follow constitutional rights and have a right to education. The right to education is going to have that curiosity in you to dig deep inside and find out what legacies Tathagat Gautam Buddha through his preachings and the Nalanda universities have given us. So thank you so much, sir. It was a wonderful talk. And आपको और थोड़ी तकलीफ देना चाहूँगी. एक और हमारे रह गए हैं, स्मिता गुड़दे, Department of Botany from AD Mahavidyalaya, Barsingi, Nagpur. Congratulations for this oral presentation, and I'm so sorry that we had just missed it out. So I welcome Smita Gurde sir, and I request Khedkar sir to do the honors. And thank you so much, sir. And last but not the least, let us hear the views of Professor Dr. Praveena Khobragade, Madam, who is principal of Dr. Baba Saheb Ambedkar College of Law. I request Dr. Subhash Somkuar sir to please introduce us to Professor Dr. Praveena Khobragade madam.
Thank you, sir.
Thank you.
Wait, sir. मी घेते आरती. Since Khedkar sir has to leave and Professor Somkuar is busy in escorting him, I take on this opportunity and I must say that I am blessed. मैडम कभी कभी जो भी चीज़ें होती है अच्छे के लिए होता है, अदरवाइज मैं ये चांस मिस कर देती आपको इंट्रोड्यूस करवाने का. सो लेट मी हैव द प्लेजर ऑफ इंट्रोड्यूसिंग यू टू द नेक्स्ट स्पीकर, डॉक्टर मिसेस प्रवीणा खोब्रागडे मैडम. शी इज इंचार्ज एंड प्रिंसिपल एंड एसोसिएट प्रोफेसर ऑफ RTM नागपुर यूनिवर्सिटी, डॉक्टर बाबा साहेब अम्बेडकर कॉलेज ऑफ लॉ नागपुर. She is a permanent faculty in RTM University, Nagpur Law University, and Madam is an associate professor and she has given her services to the university continuously past 19 years. She has handled the charge of University Law Officer for 4 years and worked as Member Secretary of University Grievances Cell and Statute Committee. Presently, she is member of Sexual Harassment Cell of the University, member of Board of Studies of Law, and she is University Recognized PhD Supervisor, and students are pursuing their PhD under her able guidance. And to her credit, there are minor research projects which are funded by ICSSR New Delhi and RTM Nagpur University. She has also contributed towards social services in the capacity as an in-charge officer, coordinator of the NSS wing of the college for 8 years. And as a commitment towards society, she is involved in conducting legal awareness camps and related activities at various schools, slums areas, nearby villages of Nagpur and also Deekshabhumi during the Dussehra festival. Madam has participated in various international, national and local level seminars and conferences, and her research articles are published in many journals, seminars and conference proceedings at state, national and international level. She has many books also to her credit, and she has delivered many guest lectures at various institutes including government organizations like National Academy of Defense Production, Indian Office Audit and Accounts, Vanamati, etc. She has worked as a member in various university level committees like Selection Committee, Placement Committee, Local Inquiry Committees, Law College Committee, Inquiry Committee. She has organized various national and state level seminars and other competitions and has been invited as an expert to express her views on legal and other issues by Nagpur as well as Mumbai Doordarshan online channels and Nagpur Akashvani. Friends, the list is endless, but I have to be very crisp, so I just hand on to Dr. Praveena Khobragade ma'am. Welcome ma'am.
Thank you very much. I pray to Lord Buddha, Shatasha Naman to Param Pujya Dr. Baba Saheb Ambedkar, the dignitaries seated on dais, Madam Mere and other dignitaries. First of all, I want to thank the organizers, Madam Mere, for inviting me and to have a dialogue with you and to share my views with you. As this is a national conference which has been organized on the innovations in science and technology for the future sustainability. This word sustainability, it is considered as a future goal, means what we have to achieve. According to me, there is a difference in this sustainability and the sustainable development. And that point of differentiation, the line of differentiation, that is very fine. The sustainable development is an idea. The idea which shows and which accepts that what is the present generation, what the resources lies with this present generation. Yes, they are that they can utilize those resources for themselves, no doubt, because at presently they are living. So this is their right to exercise and to utilize these resources, the natural resources and the other resources. But a care has to be taken by the present generation that these resources are non-renewable. And therefore, if we exhaust all these resources, then our next generation won't be getting anything. So whatever these resources available, the present generation have to utilize the resources in such a way they have to manage, they have to do that balance that the future generation will be getting those resources. Now when it comes to the rights of one individual, it has been said that right doesn't come alone. Along with the rights, there are the responsibilities, there are the duties. So if I say it is the jurisprudential point of view, so every right has a corresponding duty. So if I say, if I say that I want to exercise my right to live healthy life, to have the pollution-free environment, then in that case, I have to keep in mind that there is a one person for which there is a corresponding duty on me that I have to protect the right of that other person. So in that way, it is considered that right and the responsibility, or the right and the duty, they are the coins, or they are the sides of one single coin. So if I want to exercise my right today, I have to take care of my duty towards the next generation. So in that sense, it is the sustainable development. When I want to develop myself, then yes, I have to make the arrangement for the development, for the sustainability of my future generation. And who will be then caring for this future generation? Okay, no doubt it is the presently who are alive. They have that duty to take care that the resources are not to be exhausted, but they have to preserve those resources. So the responsibility, it is on the, majorly the responsibility is on the shoulder of the youth of this country. And the youth, which I can see, the present in this hall, yes, they have to shoulder this responsibility, and with that too, with all that, having that sense of responsibility, कि मैंने क्या करना है. Now when this is the sustainability or the sustainable development, it is for that sustainability to achieve that future goal, that everybody has to live in that peace. Peaceful life सब ही enjoy करना चाहते हैं. Now, if I take a small example of this agri-innovations. Agri-innovations, yes, as you know, that the agricultural or this field of agriculture, it is the main thing or the main component of the economy of this country. But who are the stakeholders of this industry? Not exactly, the industry status has not been given, but of this sector, the stakeholder, the farmer, his rights are not protected to what extent those are required. There are some legislations whereunder the attempts have been made to protect the rights of farmers. And that is the very recent, I can say the recent development that the state has taken that step to protect the rights of the farmers. Earlier there were some laws, the seed laws and all laws were there. But exactly the farmers' rights under this protection of plant variety and the farmers' right 2001, which has been came into effect 2005, there under some rights have been given that what plant variety or the registration for that plant variety it is to be done. And it is only after the registration the farmer can take advantage, that he can say that yes, I have the right to sow, I have the right to sell, I have the right that I can again reuse this. But when he can do so, it is only that in his name, okay, in his name, it is only after the registration. If he is having some kind of the ancestral traditional knowledge, then he can take that advantage of that traditional knowledge and he can register the seed. As it is said that the farmer, he is the annadata. He is the person who takes care of everyone. How it is? By providing food. Okay, you must have heard about the right to food. So in that, protecting the right to food, the farmer, he is playing the very major role. Now when he is protecting the rights of others, then is it not the duty of each of us, means the duty of society, including the duty of state? Because nowadays it is all the blame is given to the state, that the state is not taking care, state is not doing this and that. But the question is, the state, how this state, it is the society which has formed that legal fiction which is called as a state. So this is the society, this is the persons who are living in the society, they have that responsibility to take care of each and everyone, to take care of each other. So this sustainable development, it is nothing but one cumulative effort taken by each of us, all of us, to take care of everyone or each of us or each other. The Maitri Bhav, which has been given or which has been explained by Lord Buddha, it lies there. So this sustainability or this sustainable development, may be it for the farmer, may be it for the other person, may it be for the some kind of the responsibilities which the person has to discharge. The simple example of this, how there can be the control. ये sustainability or sustainable development, which has been referred to the developments, जो नए innovations होते हैं, and all that. If you take the example of the simple mobile, it is after certain lapse of time or some gap of time, नई quality और नया कुछ तो भी innovation mobile set में आता है. And this is all the society or this is the persons, they go to purchase for that another version of the mobile. Now what is happening when this lot many e-kachra, ये जो आ रहा है, that the earlier mobile sets, they are not yet destroyed, and these are the new versions with the new features they are coming. There is a piling of that e-waste, and there are not sufficient to dismantle or to have some kind of the arrangement. वहाँ पे कुछ भी नहीं है. Now the thing is that it is with the help of the law what can be done. It is towards that sustainable development. Why cannot be there any kind of law which will suggest or which will give some kind of the compulsion on these mobile companies that you cannot sell the new version or the new set. Before selling, you have to जो पुराना जो मोबाइल सेट है वो आपको वापस लेना है, and then again only you can have this new version. So this is how, that is to avoid that e-waste, by making the law, some kind of the control can be put on this or the manufacturing companies or the some other who are involved in this kind of the process. Few examples I can give that even as far as the agriculture is concerned. As you know that the many chemicals have been used. It is to have that production, more production. जिसको हम बोलते हैं like something that is the millets, that is the international year is celebrated, the millets. Earlier we had that practice of eating millets, okay, the traditions, but later on, then it is after the period the green revolution had come, then towards this consumption of the millet, it has been reduced. Green revolution आया है, crops का production ज़्यादा होने लगा, and it is out of that. Then to have the more production, it is the some kind of the technologies have been used, started using the new technology, the chemicals and what not to have the more production. With this, what happened? That what the fertility of the land वो तो चली गई है. Now again the time has come to regain that fertility of land. So why we need this fertility of the land? It is for that sustainable development. We have to hand over the fertile land to the next generation. So all the efforts are to be done towards achieving that, the fertile land, and how it can be achieved? That is to have that organic farming. Nowadays the trend has come of having that organic food, organic farming, taking more number of crops at a time, using less chemicals and all that. So these are the very day-to-day life instances or the things which we can do at our own end to meet that sustainable development goal. And I really appreciate the organizers, that is to organize this very important conference on this very important issue which is going to affect and which is affecting our life, and that is the sustainability. So with these few words, I put my speech on the rest. Okay, thank you, organizers. Thank you, one and all.
Thank you so much, ma'am. What a wonderful way to ignite the young minds. And it was a privilege to hear you today. And with this, dear friends, we have come to the end of this one-day national conference on innovations in science and technology for future sustainability. And I hope I have done justice to my job, and I hand over for further proceedings, that is for the vote of thanks, formal vote of thanks to my very own Dr. Rahul Kamble. And with this, I, Dr. Archana Meshram, take your leave. Have a very good day and a good evening. Thank you so much.
Thank you very much, ma'am. Please bear with me 5 minutes. It is a very hectic session, I know, but it is very important task for vote of thanks. First of all, I pay my sincere tribute to Tathagat Gautam Buddha and Bharat Ratna Dr. Babasaheb Ambedkar. On behalf of Dr. Ambedkar College and ICAR Central Institute for Cotton Research, CICR Nagpur, I extend a very warm welcome to esteemed guests on the dais, faculty members, farmers, participants, researchers, and students, and wish you a very happy International Year of Millet 2023. As the organizing secretary of this ISTFS 2023 National Conference, it's my proud privilege to thank our chief guest of the inauguration ceremony and keynote speaker, Dr. Milind Watve, ISER Pune. I take this opportunity to thank guest of honor Professor N.S. Gajbhiye, ex-Vice Chancellor DDUGU and Professor IIT Kanpur. I also thank profusely to President of the inauguration ceremony, Dr. Sudhir S. Fuljale, Secretary, Param Pujya Dr. Babasaheb Ambedkar Smarak Samiti Deekshabhumi, and President of valedictory ceremony, Dr. Mohan Khedkar, ex-Vice Chancellor, Santa Gadge Baba, Amravati University, Amravati. I wish to express my gratitude to eminent resource person for plenary talk, Dr. Suresh Umre, Dean, VNIT Nagpur, invited media persons, press representatives, participating delegates, and research scholars. On behalf of the principal of our college and the organizing committee of this national conference, I am also thankful to the farmers who are the actual farm scientists feeding people of our country. The resource persons for the farmer meets Dr. Nassim Wasnik, Dr. D.P. Gogle, Mr. Manish Khairkar, Dr. B.S. Rahile, and Dr. Pravina Khobragade ma'am. A great thanks to all. Total 325 registrations were received and about 92 research abstracts were published and 40 research scholars presented their papers in agriculture sciences, life sciences, and other sciences sections. Here also, I take opportunity to thank all the awardee members. Congratulations to all. I am very much thankful to Dr. N.M. Dongarwar, Dr. P.H. Jhaki, Dr. Devraj Saxena, Dr. Dadure for accepting our invitation and taking out time from their busy schedule to make their presence possible for this conference and share their valuable thoughts with us as a chairperson and jury members. On behalf of organizing committee, I wish to thank all the conference coordinators, members, teaching, non-teaching staffs of Dr. Ambedkar College, Deekshabhumi, Nagpur. I am also thanks to working committee of this conference for their tireless supports throughout the preparation and execution of this program. It's my moral responsibility to extend my gratitude towards ICER, CICR, Bank of India, Shankarnagar branch, Dharampet branch, Bank of Maharashtra, Surbi Chemicals, Nagpur, for providing us financial sanctions of their organizing this national conference. And also, I will be failing in my duties if I forget to acknowledge the support provided to us by the Principal Dr. Mrs. B.A. Mehere Madam and Dr. Y.G. Prasad, Director CICR. Also, thanks to all unnamed persons who have directly or indirectly helped us in organizing the ISTFS Conference 2023. Last but not the least, I am very much thankful to all my student volunteers for their untiring efforts during the conference. Thank you to all. Lastly, I wish that all the participants, stakeholders get benefited out these scientific events in developing effective collaboration and linkages among institutions. I am sure this conference will create a benchmark in discussing agri-productivity, plant environment, biodiversity, sustainability, and millets research, and will help to address the emerging challenges in agri-biosciences and will provide a roadmap for future agri-biosciences researches in India for strengthening the Indian economy. I conclude with a sincere sense of gratitude and thanks to each one of you. We are in a mission to serve the farming communities of this country. Our efforts must touch the life of the last person in the society. Let us work for humanity. Thank you. And now I request you all to raise from national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you.